शिरमन कृतज्ञ आमचे, उन्नत शिरमन कृतज्ञ आमचे. भाग्यवंत आम्ही, भाग्यवंत आम्ही, भाग्यवंत आम्ही, भाग्यवंत आम्ही. आम्ही आम्ही रुपारेलचे, आम्ही आम्ही रुपारेलचे. उन्नत शिरमन कृतज्ञ आमचे, उन्नत शिरमन कृतज्ञ आमचे. भाग्यवंत आम्ही, भाग्यवंत आम्ही, भाग्यवंत आम्ही, भाग्यवंत आम्ही. आम्ही आम्ही रुपारेलचे, आम्ही आम्ही रुपारेलचे.
मानियाळी जानियाची पंढरी तू खेळीनाची,
बेशी धरे सया वाणनसे रत्नांची.
मानियाळी जानियाची पंढरी तू खेळीनाची.
बेशी धरे सया वाणनसे रत्नांची. कल्पकुशल हस्ती तव घडे शिल्प आमुचे. ज्ञान कला खेळांगणी यशोस्तंभ आमुचे, यशोस्तंभ आमुचे. भाग्यवंत आम्ही, भाग्यवंत आम्ही, भाग्यवंत आम्ही, भाग्यवंत आम्ही. आम्ही आम्ही रुपारेलचे, आम्ही आम्ही रुपारेलचे.
तव वास्तु सदा भला तेजोमय वारसा. असू आम्ही वचनबद्ध चालविया हवसा.
वास्तु सदा भला तेजोमय वारसा.
असू आम्ही वचनबद्ध चालविया हवसा.
हो तुझे यश सदा कामन हे आमुचे. तन मन अन बुद्धी धन अर्पण हे आमुचे, अर्पण हे आमुचे. भाग्यवंत आम्ही, भाग्यवंत आम्ही, भाग्यवंत आम्ही, भाग्यवंत आम्ही. आम्ही आम्ही रुपारेलचे, आम्ही आम्ही रुपारेलचे, आम्ही आम्ही रुपारेलचे.
Good evening, all. I welcome you all on the for the second day of our lecture series, which is conducted by Physics Department of DG Ruparel College in collaboration with Marathi Vidnyan Parishad. So, I warm welcome to all the dignitaries on the dais and all the attendees for the lecture series. Now, first, let me request Honorary Secretary, Mr. Vinayak Karnik sir, to say a few words about Marathi Vidnyan Parishad.
धन्यवाद मुग्धाताई. Hello friends. I should have said hello भाग्यवंत. I just heard about this रुपारेल गीत आणि सगळेजण म्हणतात आम्ही भाग्यवंत, आम्ही भाग्यवंत. So hello all भाग्यवंत and the dignitaries present here today from Ruparel College, Marathi Vidnyan Parishad, and also today's guest speaker Dr. Ranjan Garge. Hello sir.
Hello.
Now those who were present yesterday, they have probably heard about the Festival of National Science Day. The reason behind why 28th February this day was selected as a National Science Day. For a quick recap, it was this day when Professor Chandrasekhar Venkat Raman, you all have heard or you are well aware of this famous personality. He submitted his famous research paper to the Nature magazine, which further conferred upon him the Nobel Prize in 1930. Having said this, let me introduce few awards. Those are given by Marathi Vidnyan Parishad. last few decades. Of course, few titles are constituted in the recent past. There is one Sutrim Taskar Parivar Lagu Udyojak Puraskar. This is particularly meant for the small scale industries and the startups. This particular award is of around 1 lakh rupee. Every year this award is given. Then there is Pradhyapak Manmohan Sharma Vidnyan Tantradnyan Puraskar, Science and Technology. There are two awards of this Pradhyapak Manmohan Sharma Vidnyan Tantradnyan Puraskar. One for professors from colleges in the research field for science and technology, and one is for professors who are working in the universities. Both these awards are of rupees 1 lakh. These are given every year. Then there is Vidnyan Sanshodhan Puraskar, Parshuram Baji Agashe, Vidnyan Sanshodhan Puraskar, then Leela Parshuram Agashe, Vidnyan Sanshodhan Puraskar, and Sharad Naik, Vidnyan Sanshodhan Puraskar. There are around a dozen such awards every year Marathi Vidnyan Parishad gives to the people working in various fields. There is also Manorama Bai Apte Vidnyan Prasar Puraskar. This is a particular award given for science disseminators. And there is one more recently constituted is Shri Sudhakar Uddhavrao Athale Vidnyan Prasar Puraskar, which is of 25,000 rupees. There are 3 or 4 such awards in the field of agriculture as well. Those who are working in the field of agriculture and have innovative ideas put into practice. There is Vasantrao Naik Krushi Pratishthan Puraskar. There is Bali Raja Anna Sahib Shinde Puraskar, and there is one award. I don't exactly remember that. It's a very recent award that is given in the field of environmental agriculture. There is Rashtriya Abhiyanta Din Puraskar also in the memory of Sir Mokshakundam Vishweshwara. This is also given every year by Marathi Vidnyan Parishad. There are few other programs. Yesterday there was a narration of those programs wherein the students from Parishad College are participating. We have in Marathi Vidnyan Parishad some short courses also. Those courses are 2 days courses, out of which one is in the coming 19th and 20th of February. That is on Introduction to Birding. It's a 2-day workshop, or it's a course rather. There is also a city farming course. There would be a course on Introduction to Butterflies in the recent future. There will also be a course on domestic civil works. There are a lot of activities. There are around 80 and odd activities round there being carried out in Marathi Vidnyan Parishad. I appeal all the students who are present today to join Marathi Vidnyan Parishad as a member, as a volunteer. They can subscribe to the magazine, Marathi Vidnyan Parishad's magazine also. And I request you all to visit sometimes to Marathi Vidnyan Parishad Vidnyan Bhavan, which is situated in Science Suna. But if you Google or if you browse for Marathi Vidnyan Parishad, you can visit also to our Marathi Vidnyan Parishad website, and you'll get information of lot of activities of Marathi Vidnyan Parishad. You'll get a calendar also. You can join different activities. You can join the lecture series. You can join also the science fiction. In your lecture series, there is one science fiction narration also kept for you by Captain Sunil Suri. I think let me not be a barrier in between you and the today's guest speaker, Dr. Ranjan Garge. I hand over this to the Dijiruparil College, Madam Mugdha Patki. Thank you very much. Vande Mataram. Madam, you are mute probably. You are mute. Madam, you are mute.
Sorry. Thank you, sir. Now I request our Professor Girash Sharma, sir, to introduce today's guest, Dr. Ranjan Gargi. So over to you, Sharma, sir.
Thank you, ma'am. So, uh, it's a good evening, everyone. It's a great pleasure and honor for me to introduce today's speaker, Dr. Ranjan Garge. He has completed PhD in microbiology, Bachelor of Journalism with a gold medal, and diploma in business management. He is a microbiologist, journalist, mountaineer, environmentalist, educationist, social worker, and outdoor management expert. He was director pre-IAS Training Institute, Aurangabad from 2004 to 2006 and retired as a principal, Government College of Arts and Science, Aurangabad in 2011. Currently is a director, Cosmos Outward Bound Pioneers, Aurangabad. Dr. Ranjan is in the field of management consultancy since last 15 years. He is a faculty to Department of Journalism, MGM College of Mass Communication. He is the president of Marathi Vidnyan Parishad and Indian Society for Training and Development, Aurangabad chapter. Member of Rajya Marathi Vikas Sanstha, Government of Maharashtra, and member of Vishwakosh Samiti as a chairman of the Microbiology Knowledge Corporation, Government of Maharashtra. He has authored 30 books on popular science, 5 reference volumes like Vishwakosh, curriculum books in science methodology for B.Ed and M.Ed, management and adventure. His book on mountaineering received the Best Literature Award instituted by Government of Maharashtra in 1993. Sir has been a recipient of many awards. Uh, he has been awarded a silver medal in mountaineering at the hands of 10th President of India, Dr. V.V. Giri in 1973 and Best Tracker of Maharashtra at the hands of CM of Maharashtra, Mr. Vasant Patil in 1983. He is the recipient of Best Science Propagator Award in All India Marathi Science Congress and Lokmat Gold Medal for Journalism. So, with these words and without any further delay, I will request Dr. Ranjan Garge, sir, to start the session. Thank you very much.
Thank you very much, Girash Sharma sir, for saying so high about me. I welcome the Ruparel College students and teachers, and really congratulate the Ruparel College for this very important initiative that you have taken on the eve of National Science Day. So really happy to be amongst you this evening, and I think now we shall just switch over to our topic, that is current trends in the research in biology. So I will just share the slide. So I hope you can see the slide.
Yes, sir.
Yeah.
Yes, sir. Yes, sir.
Yes. So current trends in biology research. Of course, this is in fact very vast, area, and 1 hour is not enough to cover the entire different fields in the recent advances in biology. But as you know, as a student of biology, till mid 20th century, maybe by 1950, You know that there were three areas, botany, zoology, and microbiology, which used to be included in biological science. And what exactly we used to study in those days? We used to study morphology, we used to study physiology, anatomy, and to some extent genetics. But of course, it was a turning point maybe in 1950 that the entire biological science got focused on a very small thread in a cell to which we call as a DNA. And once we actually went on exploring about DNA and RNA, about the nuclear material, more and more, the scenario of biology research actually turned in a different way. Now here we see that there are major three fields in the applied biology. The first is biology done in industrial sector, that is industrial biology. Secondly, the biology done in agricultural sector, that is agricultural biology. And the third is the biology done in medical sector on a very large scale, and that is medical biology. Of course, in these three different fields, the current trends, if we just go very quickly, in industrial biology, what we see is there is a lot of innovations going on in biopharmaceuticals. We are going to take review of everything later on. The most important thing is the 3D cell culture. We can create the 3D cell culture and then simulate how this particular kind of a drug will act upon that particular cell and what exactly would be displayed as far as the physiology is concerned. So biofuel is also one of the areas coming up very soon in industrial biology. In agricultural biology, the transgenic plants have played a huge role because now we can generate the plants of your choice, your characters, and therefore, when in those days, maybe in 50s, 1915, when we used to talk about plant pathology, that plant pathology has really come down to zero because of these transgenic plants, because of these the plants without any kind of a disease that they show. Then we also do the tissue culture is one of the advancements. Then aquaculture is a recent trend in agriculture. It is growing the plants in the water itself. Then we come to the third, and then is medical biology. In medical biology, I think there are number of challenges which are coming, year after year. For example, AIDS was a big challenge. Then came the corona, and we know that especially three fields in any kind of a medical chapter. First is diagnosis, second is epidemiology, and third part is the treatment. For example, in diagnostic, diagnostics of the diseases, the RT-PCR is again again we can say the advancement in the field of diagnosis. That is, RT is the reverse transcriptase. It is known as the reverse transcriptase technology. We are going to talk about this technology because it is also a turning point in the history of genetics. And PCR is polymerase chain reaction. These organisms, these viruses are so tiny and their DNA or RNA, unless it is multiplied, you cannot detect the presence of this nuclear material. And therefore, we have to carry on first, you can say PCR. The PCR magnifies the DNA or RNA present therein, and that is how we can identify the antigen present or the presence of that organism. And that is how this RT-PCR has become a very common word nowadays since Corona or the pandemic has come out. Secondly, epidemiology. We actually decide our strategy as to how exactly tackle that particular kind of a disease. And this strategy can only be possible unless you have the data regarding its behavior. regarding the behavior of the organism. And therefore, this particular kind of science, that is data science, and that is known as the bioinformatics, the biological information, more the information you have, more, correct strategy you can have regarding that particular disease. The third point is the treatment part. And you know that the entire world was hungry for vaccine. And we wanted vaccine very soon. My friend did already tell you vaccines cannot be made so soon, but we have made a record that within 1 year's time we could produce vaccine against for corona by 7 different methodologies. And what these methodologies are? These are
Sorry,
nothing
sir.
but the innovation. Yes,
Your voice is not clear.
voice is not clear. Okay, so 1 minute. I have made it 100%. Hello, hello.
That means other sound is there. Background sound.
I don't know, from the sound point because there is a complete calmness here. And, you cannot, hear me properly, or there is a torsion in the sound.
व्हिडिओ बंद करून काय करता येईल का बघूया का?
Video बंद करून, आता दिसतंय का? ऐकू येत नाही?
पण तुमचा आवाज हे खरखर घरघर ऐकू येते. व्हिडिओ बंद करून म्हटलं काय करता येईल का?
व्हिडिओ बंद करून आपण बघू नका. कारण इथे मी आता साऊंड जवळ 100% केलेला आहे.
काहीतरी घरघर येते ना ती जास्त आहे. तुमचा व्हिडिओ बंद करून बघूया ती घरघर कमी होते का?
हॅलो,
हा,
आता ऐकू येतोय का?
होय, आवाज येतोय. बॅकग्राऊंडचा आवाज येतोय.
बॅकग्राऊंडचा आवाज तर काय कल्पना नाही मला कशामुळे येतोय, कारण तो
आम्ही इथून शेअर करून बघू का?
बरोबर ठिकाणी शेअर करून.
नाही सर, तरी आवाज येतोच आहे.
नाही तरी माझे ते बाहेर मी काढून पाहिलं, पुन्हा 2-3 वेळा रिजॉईन करून पाहिलं तरी तो आवाज आहे.
तुमच्या हातात माईक आहे का?
हा, नाही मायक्रोफोन आहे तो.
मायक्रोफोन त्याच्याने तर येत नाही ना. मायक्रोफोन
हा जो दिसतोय तुम्हाला आता फोटो, दिस इज द मायक्रोफोन. दिस इज व्हेरी शार्प, बिकॉज गुगल मीट वरती जो प्रॉब्लेम येत
नाही,
नाही.
पण तो मायक्रोफोन काढून आवाज येतो का बघा.
नाही ते पाहिलं. हॅलो हॅलो,
तो पीसी ला अटॅच असेल ना मायक्रोफोन.
हो तो काढून
तो
पाहिला.
काढा.
हॅलो हॅलो,
थोडा कमी आहे पण
हॅलो
आहे. आवाज येतोय सर.
हॅलो, हॅलो,
हा, आवाज
हॅलो,
येतोय. हा, येतोय सर आवाज.
हॅलो.
सर आवाज येतो तुमचा. सर तुमचा व्हिडिओ बंद केला तर कदाचित व्यवस्थित येईल अजून.
हॅलो,
तुमचा सर व्हिडिओ बंद करून बघा. हा आता बोला.
हॅलो.
हो सर व्यवस्थित येतोय.
आता येतोय का आवाज?
हो हो.
That's good somehow. Okay, okay. So now will you share from that side only and let us continue. Hello. Hello, Sugar, can you hear me?
शेअरिंग ऑफ करू का?
नाही नाही तिकडनं चालू करा
ओके ठीक
व्हिडिओ.
आहे. तुम्ही सांगा नेक्स्ट जायचं असेल तर नेक्स्ट
हो.
बोला, मग मी नेक्स्ट.
Yes, next. Next slide, please. Next slide. Next slide. Yeah. Okay. Yeah. Now can you hear me properly?
Yes sir. Yes sir.
Yeah. Okay. So now we shall yes one slide. Go back one slide. Yeah. So we were talking about the advances in industrial biology, then agricultural biology, and medical biology. And I was talking about the RT-PCR, that is the reverse transcriptase polymerase chain reaction, as one of the advancements. I was talking about the bioinformatics is the another advancement. I was talking about the vaccine preparation methodology, that is in vaccinology. We have gone far ahead and we have 7 different methods by which we could prepare the vaccine for corona. And that too, mRNA vaccine is supposed to be the miracle in the field of molecular biology. Organ cloning is again one of the coming up areas. The most interesting is pod babies. We know about the in vitro fertilization, but pod baby is just one step ahead. That the baby born in the glass test tube itself, that also these are also the possibilities in future. Biobricks is the another area which I'm going to talk in details. And therefore, cell biology, stem cell research, and gene therapy, they ultimately are going to be the center point as far as the treatment part for any kind of disorder is concerned. Maybe cancer, maybe corona, maybe AIDS, maybe any kind of a disorder which is really difficult to cure. Next slide, please. Now there are applied biology branches, other branches, and they're also equally important. For example, water biology is a big area because aquatic life, there are a lot of research going on in aquatic life. Sewage biology, bioremediation is a new area which is coming up where we are now going to treat the sewage with biological elements and not by chemistry. Then food biology, milk and milk products biology. For example, yogurt, for example, the lactobacillus culture, and all these kinds of probiotics is going to be the second point, in the 21st century. Then soil biology, basically related to agriculture. Air biology related to air pollution. Then population biology, population dynamics throughout the world has become very important, and therefore there are a lot of statistical tools nowadays being used for population biology, and then health biology. So these are number of areas where actually we see that the upcoming areas in the field of biology are coming very fast. Next slide, please. Now here what we see, next slide. Yeah. Now, initially, as I was telling you, how the whole journey went on. See, botany, zoology, and microbiology, those these were the key elements which we used to study in biology. See, this biology went on in the interdisciplinary approach. And this approach was basically the chemistry and biology became biochemistry. Then physics came in, and therefore biophysics became a very important branch. Then genetics and genetic engineering, then DNA recombinant technology. Then we went ahead and therefore we now call to all these genetic engineering technology as a biotechnology. Then genomics. From genomics we jumped on to proteomics. From proteomics to transproteomics as a chip technology. And now we know that we are in the silico drug design also. So see, this particular pyramid tells us that how exactly from simplicity to complexity this entire research went on when it went on in hand with other areas in the field of science. Next slide, please. Before we actually go to the three to four technologies which I am going to talk about, we must see what are the six fundamental discoveries which laid the foundation of modern biology. Because under these six discoveries were there, it was impossible, for the scientists to go to the modern science as on today, what we call it as. Next slide, please. Now, the first fundamental discovery. First fundamental discovery was the transformation. See, bacterial transformation is the process of horizontal gene transfer by which some bacteria take up the foreign genetic material from the environment. And therefore, it will observe that the Staphylococcus pneumonia, the organism which causes pneumonia, this particular organism, very interestingly, was getting converted from virulent to non-virulent and from non-virulent back to virulent. And therefore, these scientists, especially the Frederick Griffith and the Oswald Avery, they studied that how exactly this organism changes itself from virulent to non-virulent strain. And they observed that in 1924, they found that DNA is the transforming principle. Now I'm talking of 1944, when the DNA structure was also not known. So before we knew the DNA structure, the Oswald Avery and these two scientists basically could discover that this DNA thread basically transforms some characteristics by way of which this particular organism shows, gives two, ways as far as virulence is concerned, virulent and non-virulent. So this transformation was a very important kind of a landmark, first landmark in the history of modern biology. After 1944, next slide please, the second phenomenon, that was the another turning point was in 1948. In Escherichia coli, a small organism, they found that two organisms, they actually show the process of conjugation. They conjugate. But Edward Stepney and Joshua Lederberg, they show that when this conjugation takes place, the organism having F factor, fertility factor, transfers this fertility factor to the another E. coli strain which doesn't have fertility factor. So it was very interesting that there is a polarity in microorganisms that was for the first time observed, that there is F plus strain and there is F minus strain. There is F plus E. coli and there is F minus E. coli. So the conjugation is the process of transfer of this F factor from F plus strain of E. coli to F minus strain of E. coli. And this actually gives a clue that we can transfer the characteristics from one particular cell to to another one. And you know that this conjugation method, this conjugation discovery actually gave the Nobel Prize, which was shared between Lederberg and Tatum, and that in 1958. So I think this was very important point, and they were trying to see that how does DNA and what sort of transformations this DNA can bring about from cell to cell contact. Next slide, please. Now, in the next slide, the third phenomena we are going to see. Next slide, please. The third phenomena is known as transduction. Yes, will you please change the slide? Next, yeah. The third phenomena is transduction. Now, what is transduction? They found that the salmonella, the organism which causes typhoid, in 1952, they found that salmonella gets infected with a virus, and that is known as bacteriophage. And very interestingly, this bacteriophage gets attached with the DNA of salmonella, and then after some time, through the process of lysis, it again, comes out of the salmonella cell. Let me tell you that this particular transduction phenomena ultimately has given rise to the CRISPR technology, which is going to be a very important landmark in the history of biology in 21st century. You must have heard about CRISPR, and we are also going to talk about CRISPR technology later on. So, why I'm saying that these are basic and fundamental discoveries? Because once the scientists knew that how the DNA plays in between two cells, two adjacent cells, and how the transformations takes place by these three different ways, that is transformation, conjugation, and transduction, then of course they could really understand the genetics, microbial genetics. Microbial genetics are very important to be understood first because DNA is a simple part of nucleus, and therefore once you understand DNA, it was easier to understand the complicated part of DNA, that is chromosomes, that we see in the plant cells and the human and the animal cells. Next slide, please. The fourth important discovery, I would say this was a turning point in the history of biology, and that took place in 1953. 25th April 1953. You know that ultimately the structure of DNA was known, and it was found to be the double-stranded DNA, and in 1962, the Nobel Prize was shared between three scientists. First was the Francis Crick, second was the James Watson, and the third was Wilkins. There was a fourth member also, and she was the Rosalind Franklin. Rosalind Franklin has really contributed more than 60% in this particular discovery. But but she died out of cancer before 1962, and therefore she was not awarded the Nobel. But she was an expert in X-ray crystallography, and the structure of DNA was for the first time observed through X-ray crystallography by Rosalind Franklin. So the point is, the structure of DNA ultimately was discovered in 1953. After 1953, the most important discovery that we are going to see, that is discovery number 5, which is a basic fundamental discovery. Next slide, please. And that particular discovery is about the presence of plasmids. Till 1952, nobody knew that any single cell contains a double-stranded DNA molecule, which is other than the nuclear material in the or in bacteria and in yeast. And this was discovered by William James and the Joshua Lederberg. So Lederberg was a great man. As we saw, he invented the process of transduction, at the same time process of conjugation, and at the same time he was the discoverer of even the plasmids. See, in the red color, you see the DNA part of it in the bacterial cell, and you find the blue part, and these are the plasmids. You can have more than one plasmid. These are independently available double-stranded DNAs, and they can multiply, they can replicate independently. But when in 1952, these two scientists discovered the plasmid, presence of plasmid, such kind of a DNA structure, apart from nucleus in a bacterial cell. It was that way neglected, and nobody thought that this is that important. But we find that exactly 1970s, plasmids gain a great prominence. And let me tell you, the another discovery actually is based on the plasmids only, and that is the biobricks. Biobricks technology that is being used, actually that is yet to be used in fact in 21st century, is basically dependent on the plasmids only. So the transduction and the discovery of plasmid, these are the two very important fundamental discoveries which gave rise to CRISPR technology and the, as I told you, biobricks technology in future. Next slide, please. The sixth and the last important discovery, basic discovery, was the Human Genome Project. Friends, this Human Genome Project was a great achievement throughout the world, and you know that it actually, this project was picked up in 1984 by US government, and it was formally launched in 1990, and it was completed in record time. It was actually declared complete on April 14, 2003. And the level of complete genome, all 23 chromosomes were mapped by May 2021. So mapping of chromosomes, I think, is a very fundamental discovery, and because of which, in future, we are now approaching towards the gene therapy as a common therapy, which may find maybe by 2025 to 2030. So James Watson, the one who got Nobel for the discovery of DNA structure, that James Watson, now an old man, he assumed the role of overall project head and the director of US National Institute of Health. He was the director of this Human Genome Project. And ultimately, in record time, as I told you, we could map all the 23 chromosomes, 22 chromosomal pair, and the 23rd chromosomal pair as the sex chromosome. So these six discoveries, with the background of this, or having understood these six discoveries, now we shall enter into the modern practice. So next slide. Now here's a biology clock. And this biology clock actually tells you how these, of course, I have sorted out these 12 very, very important areas of the current trends in the discovery of the research of biology. The first is genomics, bioinformatics, then proteomics, then transproteomics, then the global warming and biodiversity, a very important and current area of research in the biology, then synthetic biology, then in silico biology, then CRISPR-Cas9 and gene editing biology, then CCR5 gene and HIV. This is again a gene editing biology related to HIV. Then astrobiology, that is extraterrestrial biology, finding a life on the other planets, and then origin of life. See, origin of life has become a hot area of research today because everybody is now curious to understand what exactly and how exactly the first life came on this planet. And then the biotechnology. So these are the 12 areas where we find that the current test in biology exists. Of course, it is very difficult to take the review of all these 12 areas, but important one we shall see one by one. So next slide, please. Now, here we come to the biotechnology. These are the five different biotechnology areas. See, white biotechnology, it is known as white or gray biotechnology. Whatever biotechnology, see now the word is changed. It is from biology to biotechnology. That means you are using that biology in the form of a technique. So biological technology being used in the industrial processes. Fermentation industry, dairy industry, so the probiotic industries, the pharmaceutical industries, they are all the industries which fall in basically white and gray biotechnology. The second area of biotechnology is green biotechnology, and that is basically the technology, biotechnology applicable to the agricultural process. As I told you, the tissue culture technology and the different kinds of, the varieties, the crop varieties that we are now getting, all these are through the green biotechnology. The third biotechnology area is in silico biotechnology, and that is nothing but application of the computer science, applied mathematics, and statistics to address the biological problems. And I think this intermingling of mathematics and statistics with biology has increased to such an extent that there are many models. When you do the mathematical modeling of a biological entity, this in silico biotechnology comes in picture. Then we come to the fourth area, and it is blue technology. That is basically the technology applied to marine life and the aquatic processes. And then we come to the fifth one, that is RAID biotechnology. RAID biotechnology is the technology applicable to the medical processes. In medical processes, a lot of things are happening, right from cloning to IVF to so many things, and we are definitely going to see few examples. So cancer is one problem, and so many other things. So, white or gray biotechnology, green biotechnology, in silico biotechnology, blue technology, and red technology, these are five major areas of biotechnology that are ahead of us in 21st century. Next slide, please. Uh, Now, here we see the other applied branches. See here the cell culture. Cell culture is, again, as I told you, the hot area. There are three hot areas. Um, First is the cell culture, second is the monoclonal antibody. So basically, all these autoimmune diseases are now going to be treated in future, or maybe the cancer treatment is going to be based on the monoclonal antibody. That means you develop the antibody with known receptors, and that is how you actually counterattack that particular kind of disorder. In cell culture, in plant cell culture tissue, gene transfer into animal organs is also possible through the cell culture technology. And the third technology is the molecular biology. So, cell culture, monoclonal antibodies, and the molecular biology. In molecular biology, you have again three areas. The first is the DNA technology itself. So what do you mean by DNA technology? For example, bank of DNA. So DNAs will be preserved, RNAs are preserved, special and specific proteins which have a value addition, they are actually preserved the complete map of human genome. So all these activities, they all come under the DNA technology. Second is genetic engineering. In genetic engineering, there are two areas. First is the synthesis of new proteins. That means new types of animal and plants can be generated by synthesis of new proteins. New types of foods can be generated by synthesis of new proteins. New antibiotics can be generated through the synthesis of new proteins. This is area ahead. The second in genetic engineering is cloning, and that is the mass production of the human proteins through cloning. That is possible. Then the resource bank for the the rare chemicals. This also is possible through cloning, and gene therapy is also possible through cloning. And therefore, most of the diseases like maybe the diabetes, as we can say, can be treated through gene therapy. And then we come to the third area, and that is the use of the cases. There is synthesis of the specific DNA probes. This is very important, which can localize the genetic disorder. So localizing the genetic disorder through the genetic DNA probes is possible through the research technology. So DNA technology, genetic engineering technology, and research technology, they all come under the molecular biology. So these are some of the areas which we expect in future to deal with. Yeah, next slide, please. Biotechnology, of course, this is for you basic students. You all know that application of the scientific and engineering principles to biological processes to provide goods and services and generate business out of it. It is known as biotechnology. Next slide, please. In the next slide, what you see is through this biotechnology technique, you can now not only produce antibiotics, but you also can get the probiotics. Probiotics is nothing but actually making use of the friendly microorganisms like lactobacilli for the treatment part of it, for the treatment part of it. So this is how the antibiotics to probiotics that we are really going to change in near future. We have actually started taking probiotics nowadays. Biological extracts, liver extracts, pancreas extracts. So these extracts are also going to be very widely used in the because they have a medicinal value. Next slide. In the next slide, what we see is the genetics to genomics to proteomics to transproteomics. See, this is the way you are going to travel now. Classical genetics was like this, as you know, as a student of biology. That is DNA, then DNA replicates, and that is the central dogma. Then passing information from DNA to RNA to protein, and that is how the protein synthesis takes place. So this is the classic genetics or classical genetics that you must have studied. Next, now when you talk of genomics, genomics is a science where you make the mutations. Making mutations, the natural process of exchange of genetic information, mapping of chromosomes, the operon model of a gene function, then breeding techniques, developing industrially important strains, then genetic engineering of that is basically RDNA technology. So these are the technologies which come under the genomics. That means in genomics, you are basically bringing out the mutations through the breeding techniques and all that, and you are actually getting or trying to get or trying to develop industrially important strains. Next slide. From genomics to proteomics. Now, what is proteomics? Now, in genomics, you actually ask, the genes to undergo the protein synthesis and the wanted or important proteins are produced. But in proteomics, see, understanding different proteins made by the different genes, you first understand that what kind of gene produces what kind of protein. And when and how are the genes activated? That also you study. So that you activate a desired gene to get a desired protein. And so characteristics and the functions of these proteins are to be identified, and then such proteins are then directly injected or used as a technology. So instead of actually stimulating the gene to produce a protein, here you are actually identifying that which gene after activation, what kind of protein is being produced, and that protein itself is being used in this technology. Next slide, please. After proteomics, in proteomics itself, what way these proteins are related to human life, that is also studied. Every cell has potential to make all the proteins because you know that every cell has a DNA. And so you have to exploit every cell to make the desired protein. Now, how does the cell make desired proteins only and not the unwanted one? You know that unwanted protein making actually results into many disorders. And therefore, here exactly you want the cell to make a desired protein. And last question is, can protein be used for human treatment? So this is all being studied very carefully, and then proteomics is applied. Next slide. Then transproteomics. Now we go beyond the proteomics now, and that is known as the chip technology. Affymetrix is an American company, and it has come up with the genetic chips in the form of a glass slide. It's a simple glass slide, and the glass slide is coated with DNA. Um, with processing the mRNA sequence, there is a specific mRNA sequence on that particular glass, and different cells can be tested accordingly. And so, two chips have been developed like that. One recognizes about 60,000 messenger RNA molecules, and there is another chip that has been developed which recognizes the 1700 mRNA molecules. So, next slide, please. Now, what is the application of this chip technology? The chip technology basically is used for the that is the astrobiology. That is trying to find the existence of life on another planet. See here, the small dots that are seen on this slide, they are actually the pores where fluids and the chemicals can be mixed or samples can be collected for testing. Tiny channels, only clearly visible under the microscope, form pathway between the pores. Many chemical and biological processes previously conducted on large pieces of laboratory equipment can now be performed on these small glass or plastic plates. First, this is Monaco and her research team at NASA, the Marshall Space Flight Center at Allah, and these are customizing the chips to be used for many space applications, such as monitoring microbes inside spacecraft and detecting life on the other planet and so on and so forth. So, lab on a chip by Dr. Lisa Monaco. So this is entirely a new area. So you have actually brought the entire laboratory on a glass or a plastic plate as such. Next slide, please. This is very interesting kind of cartoon. So here the this particular transgenic the vegetables, what they say to the professor They say, Professor, you want to discuss certain flaws, we want to discuss certain flaws in your scientific theory. See, now the transgenic, plants themselves have become smarter, and therefore the scientists say, "Oh, that's it. We have taken the genetic engineering too far." So now genetic engineering has become a very interesting thing because the transgenic plants and transgenic animals may tell you what is the flaw in your genetic engineering technology. Next level. Uh, Now come to the synthetic biology. Synthetic biology is a new term that is coming up. Synthetic biology is an engineering counterpart to biology. Whether it is building a novel genome or editing an existing one to add a new function or to remove the previous function, the problematic pathway can be removed, and the idea is to improve the performance. Synthetic biology got its first push in the year 2000 through the toggle switch that let researchers turn the gene on and off. So when the scientists acquired the technique of turning on and off, turning the gene on and off, actually it became the beginning of synthetic biology. An oscillator that switches a gene on or off depends on the state of another specified gene or a protein. So these are the basic concepts of synthetic biology. Next slide. Synthetic biology works with biology, systems biology, bioinformatics, mathematics, chemistry, biotechnology, and engineering. Next slide, please. Basically, in this particular kind of synthetic biology, a very important paradigm shift has taken place. And that is, instead of the body parts, now we shall be talking of the genetic parts. And therefore, instead of organ transplant, we shall be transplanting the bricks, and these are the bio bricks. So bio bricks will be used in these transplants. And either, as I told you, removing the unwanted part or putting the wanted part therein. So all these kinds of activities would be done through the bio bricks rather than actually hampering that particular organ. Next slide. There is a terminology known as the iGen. It is International Genetically Engineered Machine. This is a registry for the standard biological parts, and it is growing collection of the genetic parts that can be mixed and matched to build synthetic biology devices and systems. So you actually create the nucleotide sequence. That nucleotide sequence is of some use in some kind of an activity, maybe fermentation activity, maybe whatever it is, maybe cancer treatment activity. Then that particular kind of a sequence is to be called as a standard biological part. And such standard biological parts are registered under this iGen. So teams and other researchers are encouraged to submit their own biological parts to this registry and to help this resource stay current and grow year to year. Today there are about 2,000 parts, standard biological parts available in the repository. Next slide. What are these biological parts? The biological part is a sequence of DNA that encodes for a biological function. For example, it can be a promoter or a protein coding sequence. It is simplest and basic part, and it is performing a single functional unit that cannot be divided further into smaller functional units. So it is one function being either promoted by the promoter or being synthesized through the protein coding sequence. Next. But ultimately it is in the form of a biobrick. The first attempt to create a list of standard biological parts was made in 1996, And this team introduced a cloning strategy for assembly of short DNA fragments. These are short DNA fragments. In 1999, Arkin and Andy, they realized that the heterogeneous elements that made up the genetic circuit were lacking standards, and so they proposed a list of standard biological parts. And that is how the standard biological part concept came into existence in 1999 in the form of biobricks. Next. Now, these biobricks, how do they look and what exactly they do? Biobricks is nothing but a genetic manipulation of the plasmids. Most biobrick parts, see the black part that you see is a biobrick part, and the red part, the red outer lining is actually the backbone of that particular what you can say plasmid. So most biobrick parts in the registry are maintained and propagated on plasmids. And therefore, this BBA B0015 is a sequence. And this sequence has been inserted into that particular plasmid. And this particular device now uh, can be used anywhere. So this now goes to a repository, and this repository, whenever is required by somebody to do the genetic manipulation, this biobrick plasmid can be inserted into whatever cell you want to. And that is how this concept works. So biobrick plasmid backbone with some biobrick part. That is biobrick part is black one, is shown by the blue lining, and the red lining is the plasmid backbone. And so this is a very recent concept in the research in biology that is coming nowadays. Next. Biobricks were described and introduced by Tom Knight at the Massachusetts Institute of Technology in 2003. And as I told you, these are the synthetic biological circuits that together have a unique function. For example, the biological or the biobrick part include coding sequences, promoters, ribosomal binding sites for the terminators. Next slide. The next thing, in silico research. In silico research is basically going to help the pharmaceuticals in making drugs, cancer research, and stem cell research. Especially 3D cell culture is used to study the body metabolism in this in silico research. Next slide, please. Uh, you can make spheroids in this particular technology, that is the spherical cellular aggregate. You can actually make the organoids. That is, organoids are the tiny self-organized three-dimensional tissue cultures that are derived from stem cells. And such cultures can be crafted to replicate much of the complexity of the organ. So, if you want to really see what happens or what is the effect of so and so drug on a specific organ, then that organ can be simulated in the form of an organoid and can be tested. Organ on chip. This is new technology. Liver on chip or lungs on chip, that is possible. And this is all possible through, as I told you, 3D bioprinting technology. Next. In silico drug design. Now, see, if this drug is to be designed in a specific way, now there are chips available, and in the next slide we shall see what are the different characteristics they are manipulated for this drug. Next slide, please. So here, a chemical structure that you want, molecular modeling of that drug that you want, homology modeling of that drug that you want, binding site predictions that you want. You want where exactly that particular drug to dock. Then the screening, then legend design. then binding free energy estimation, then the quantitative structure activity relationship, and the absorption, distribution, metabolism, and extraction of that particular drug. So all these kind of data can be manipulated through that particular chip. And that is a very recent kind of a technology, and therefore I always say that this entire pharmaceutical industry can be accommodated in maybe a 10 by 10 room, and you can get the drugs as per your desire and design through this cheap technology. Next. These are the chips which are actually implanted inside the brain, and the functions can be monitored by electrical activity of a small section of the brain, mathematically interpreting the data and relaying the new information to the action performing device. Now, this is possible. This is the head tumor chip that is being implanted in the brain. So this chip technology will tell you the entire biological activity or biological changes or anything that is going on inside the brain through the electrical signals. Next slide. And now we come to the CRISPR technology. That is, that the clustered regularly interspaced short palindromic repeats. These are the repeats of the nucleic acid. So, genomic editing is a group of technologies that give scientists the ability to change the organism DNA. And these technologies allow genetic material to be added, removed, or altered at a particular location in the genome. So, this is the gene editing technology. How it works, next slide, please. In CRISPR technology, it was adapted from the naturally occurring genome editing system in bacteria. See, the bacteria capture snippets of DNA from invading virus. You know that in transduction, the virus actually infects the bacteria. But when the virus enters into the bacterial cell, the bacteria captures the snippet of DNA from the virus itself and uses them as or to create DNA segments, and these DNA segments are known as the CRISPR array. The CRISPR array allows the bacteria to remember the viruses. If the virus attacks again, the bacteria produces the RNA segment from the CRISPR array to target the virus and kills it. This particular CRISPR array is very typical. This is palindromic. Palindromic means this particular nucleotide sequence is same when you read from both the sides. And these are the repetitive sequences coming through the virus into the bacterial cell, and they are now being used for cutting the DNA. That is, it has become a chemical scissor, and that is how you can edit the DNA. We'll see the picture. Next slide. Next slide. Now, CRISPR-Cas9 is a protein was adapted from a naturally occurring genome editing system in E. coli. In 1987, for the first time in E. coli, this particular kind of a palindromic repeat was observed, and it was therefore called as the CRISPR. Next slide. Uh, Next slide, please. Yeah, now here we will see how the CRISPR works. Now suppose the cell is trans-infected with an enzyme complex containing, see, this is the small circle that you see on a chromosome where there is a defect, okay? Now what you do is you insert three things. The first is the guide molecule, second is the healthy DNA copy of that particular molecule, and the DNA cutting enzyme. The Cas9 is the DNA cutting enzyme. Now, in the second figure that you see is a specially designed synthetic guide molecule finds the target DNA strand. So this is the bluish one is a guide molecule. This guide molecule actually finds this particular red part where there is a defect. Okay, this guide molecule actually traces out this red part which is defective. Now, what happens next is once the site is identified, when this red site is identified, the DNA cutting enzyme, that is, as I told you, Cas9 protein, it actually cuts that particular red part as it is seen in the picture. And the defective DNA strand is removed. And in the last picture that you see is the defective DNA strand is now replaced by this green healthy DNA strand. So first you identify with the blue one the defective part, then you cut the red part, and then the healthy DNA part is replaced. And this is therefore is a type of editing that is done in the CRISPR technology. Next slide, please. Between 2014 and 2015, scientists reported the successful use of this CRISPR-Cas9 in mice to eliminate muscular dystrophy and cure a rat liver disease and to make human cells immune to HIV. See, very interesting kind of result through this CRISPR technology that we are getting. We are editing the DNA through this. Next slide. Next slide. So CRISPR has been used to experiment with gene editing basically in mosquito. Gene editing in mosquito to reduce the spread of malaria. For engineering agriculture to withstand climate change and in human clinical trials to treat a range of diseases from cancer to the rare protein disorder of the nerve cells and the organs. So in mosquito, you can edit the gene. In agricultural engineering, you can edit the gene which can withstand the climate change. So all these kinds of possibilities are there by the use of this CRISPR. Next. And now we come to the last technology, and that is CCR5 technology. See, we have been trying to find out the cure for HIV. We have remdesivir, we have so many other, kinds of medicines, but can there be a cure, genetic cure? And it was observed that there is a very special kind of a gene known as CCR5 gene, and those individuals who do not have this CCR5 gene they are found to be immune to this HIV infection. They don't show HIV infection. So CCR5 usually codes for a receptor on the surface of white blood cells, and it plays a role of normal immune responses. And we exactly now see in the next figure as to how this CCR works. Next slide. Yes. So coming to the first slide, that is first part, the left part of it. See here, this particular, the HIV virus is getting attached to the surface of a white blood cell. Now these are the receptors. Now see, on white blood cell surface, there are two receptors. One is basically known as CD4 and other is known as CCR5. Now CCR5 and CD4 both are required. CD4 and CCR5 both are required for this particular docking. This is known as docking. The virus is getting docked with the WBC. So for docking purpose, these both CCR5 receptors and CD4 receptors are required. Then only the infection is possible. Then only this particular RNA goes into the cell. But now, suppose you actually make the CCR5 receptor defective by delta 32 mutation. See what has happened. This particular CCR5 has become smaller and it has lost itself and gone into the cell. Now there is no CCR5 available on the WBC cell surface. And therefore now the docking has taken place only with CD4. But this single CD4 docking will not allow this particular virus to enter into the WBC or lymphocyte. And therefore, by mutating the CCR5, you can stop this particular virus from actually entering into the white blood cell or the uh, lymphocyte. So this particular technology is now developing very fast, where you can actually mutate the CCR5 and stop this particular HIV infection. So this is very important, I think, landmark in the history of biology, in the history of medical biology. So CCR5 delta 32 hampers the HIV's ability to infiltrate immune cells. The mutation causes the CCR5 receptor on the outside of the cell to develop smaller than unusual and no longer sit outside the cell, and that is how you get it. Next slide. So next slide, and we come to an end. If somebody really wants to pursue for the advancements in biological research, then these are the different areas or the different knowledge areas. First is biology, second is the physionics, third is the cellomics, then biotech, then evolution, then infotech, then ontology, then proteomics, then molecular modeling, then mathematics, then metabiolomics, then transproteomics, then genomics, and the statistics. So it is therefore a multidisciplinary approach that is required. This is one of my books that has been recently come available on Amazon, and which talks about two sides of the same coin. Here you will come across number of different discoveries in 6 to 7 different areas of science, where there are two sides and how the paradigms got shifted. So next slide. So thank you very much for the uh, patient hearing. Though there were some technical flaws, you really bear that, and that is how I conclude. Now I think we can go for the question and answer session. Thank you very much.
Yes, now session is open for questions. So you can type in the chat box or you can ask orally.
Yes, sir.
Ah, yes.
Please switch on your camera.
Okay. Yeah. Yes. So
So I think there are no questions now.
I think it takes some time to questions to be generated.
Yes, sir. Yes.
So that there are two possibilities. One is either you understood everything or you understood nothing. There are no questions.
Positive. Let us be positive.
Let
Every
us be positive.
all the students, those who were in the auditorium, virtual
Yes.
auditorium, have understood this subject. I feel let us conclude.
Okay. Yes. Thank you.
Okay. So On behalf of DG Ruparel College and Marathi Vidnyan Parishad, I'd like to extend vote of thanks to Dr. Ranjan Garge sir for this informative and enlightening lecture. Sir has explained the applications of biology in major fields, right from industrial to medical. And sir has also given an overview of 6 fundamental discoveries, which leads to the advanced biology which we have today. And sir has also discussed about biological clock, which I found to be really innovative, in which he discussed 12 important areas, right from origin of life to biotechnology. So I'm sure all will be benefited through this session. And once again, thank you, sir. Thank you all.
Thank you very much. Thank you, Praneeth.
Thank you very much, Dr. Ranjan Garge.
Thank you
Thank you, sir.
very much. Thank you.
Thank you,
It
sir.
was really interesting. So many subjects were covered. So
Yeah.
I mean, we are from physics, most of us. So, but still, we could understand so many things. I mean, there is a lot that is going in this particular
Yeah.
field.
Yeah.
So, I mean, about that chip technology and all.
Yeah.
This was really interesting.
Yeah. Synthetic biology is going to rule the world tomorrow.
Yeah.
Yes.
So thank you.
Yes, thank you. Vande Mataram, Dr. Ranjan.
So, yes, we can keep now.
Madam,
Okay.
बोला सर.
मुग्धा मॅडम, हा
बोला ना.
ओव्हरऑल पाहताना थोडस असं दिसत आहे
हा
की
हा.
विद्यार्थ्यांची उपस्थिती
आज एकदमच कमी
एकदमच कमी
होती.
झाली.
हा.
ती काय लिमिट टेंड्स टू